This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 204, recorded on October 19th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. This episode of TWiV is sponsored by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Is it pouring out there?
It's raining off and on, yeah.
It's been pouring here all day.
It's pretty miserable.
It's like, it is a little mild. It's almost 70, but torrential rain.
Yeah.
Cloudy skies. How have you been, Alan?
Oh, pretty well. I've been missing TWiV. There's this hole in my Friday afternoons that I just sort of cast about and say, gee, I ought to be doing something.
Just try and give you guys a break, you know.
Yeah.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
How are you?
Hello.
How's
Fine.
the weather?
It's nice. Partly cloudy, 54 degrees, which is 12 Celsius.
No rain.
No rain.
So is it still pretty dry there? Or
Oh, no, we've had
You're
rain.
okay?
And in fact, there was rain earlier today.
Yeah.
But
Probably our rain came from you, right?
often it does. I'm not sure about what you're having right now, but yeah, we had a really hard rainstorm part of last night. That's true.
Well, my clouds say Ann Arbor on them, so
Then they came
must
from
come
us.
from you.
Yeah.
We have a special guest today. Gosh, he's probably been on 3 or 4 or 5 times. We have a new coronavirus outbreak or isolation, and when there's a corona, you know who to call. Matt Frieman.
Hello, guys and lady. How are you guys?
Fine.
How does it go? Who are you going to call? Ghostbusters. So who are you going to call? Matt Frieman.
Yeah.
There you go. I'll take that. That's okay.
So are you still an assistant professor at the University of Maryland, Baltimore?
Yes, I am.
You
I've
can
been here 3 years now.
Okay, 3 more years, then you get promoted, right?
We actually have 10 before we have to go for tenure, but we can be associate before we get tenure. So
Okay.
a couple of years, and I'll go up to associate, hopefully.
All right. How's life in general? Good?
Life is good.
Yeah. I heard you have 5 R01s.
No, I reviewed 5 R1, the study section. No, just one. So we're happy with the one. I'm not. We're busy as we can be.
So your lab is fully stocked now and running, is that right?
Yeah, we're all good to go. The BL3 works and is hopping, and I have 3 students. One just graduated, which my first student graduated, which is
good.
we're very happy with. A great project. Paper just came out in JV a couple weeks ago. If anyone wants to look it up, and
Is that the one you sent us?
no, that was a different one
Okay, it's not the one with
with Eric.
Eric. No, okay.
Yeah, no, this was on SARS and host responses to SARS that mediate severe lung disease. So
Right.
Carly Page is the grad student, so she did a great PhD, great job.
So you had a student finish in 3 years in your
Well,
lab?
Yeah, I know. It's a different story. So she was actually in Donna Farber's lab, who's now at Columbia. with
Okay.
Vince. And so she was there for 2 years. And then when Donna moved from Maryland to Columbia, Carly stayed and joined my lab.
Got it.
It was great. She was actually trained as a influenza immunologist doing memory in Donna's lab. So she switched right over to SARS and could do mice and pathogenesis and flow cytometry, and we taught her how to do everything else. So it was great. Good combination.
So what is she doing now?
So she's now looking for a postdoc, deciding where to go. She potentially Chicago, potentially staying here. So we'll see where she goes. But she's good. She's very good. It was a really good experience for my first student to kind of be a step mentor rather than a primary.
Right. So how many other students do you have?
So 2 other students.
And postdoc?
And we just got one postdoc started a couple weeks ago.
Great.
So we're going. It's you have to spend the money to make the money, I guess. So
You do?
we're trying to use what we have, build up papers, and get grants out.
Great. Yeah, don't be cheap.
No, that's what I've definitely seen. You have to put the reagents in and the time in and spend the grant money that you have to be able to get more grants.
I even spend grant money I don't have.
I think that's common as well.
I used to have a big problem with overdrafting here years and years ago. I don't know why. And lately it's fine because we have these accounting things that prevent you from overspending. But the other night I had a dream that I was, $500,000 in the red.
Ooh.
I woke up and I say, wait a minute. I thought those days were gone.
You'll have to start doing the podcast from Starbucks or someplace with free Wi-Fi instead of...
That's right.
Okay, we got you here, Matt, to talk about this new corona. But before we dive into that, we had 2 email follow-ups from previous episodes, recent episodes. The first one is Tom, who writes, "Hi, Vince. In case this didn't make it in the news in the city, I thought you might be interested in this report of hantavirus in the Adirondacks. Looks like a wooden lean-to was the scene of the crime again." Well, Tom, every news makes it to the city, but thank you for sending that. This is a guy, actually a professor from Stony Brook, who was camping in a lean-to in August. He got bitten by a mouse, and then in September developed a respiratory illness. I don't see here that it was actually diagnosed as a hanta illness. But I have to say that I doubt he got it from being bitten by the mouse.
Yeah.
It's a respiratory transmitted virus, right? So I don't think if a virus found itself in the blood, it would know what to do. What do you think, Matt? What do you think, Matt?
Yeah, I don't know. I mean, I think that's probably true. It wasn't. I mean, the one in out in the west was more droppings of mice, right? Isn't that what they thought?
Yeah, I'm sure that's what this is. He was in this lean-to and...
and his wife swept it out during their visit.
Ah, there you go.
Oh, yep. Okay.
A primitive 3-sided wood-hewn shelter. Yeah, I'm sure that's what it is. And that's a good argument not to sweep out your primitive lean-to. Okay.
Pitch a tent next to it.
Thanks for
Pitch a tent, exactly. I'll remember that. But who knows? Maybe the ground there was positive as well, so can't be sure.
Okay, the next one is from Jeffrey, who writes, "Dear cast of thousands, normally you speak far enough over my head that I just sit back and absorb information at the feet of the masters. Occasionally, I admit, I fall into an information overload-induced coma, fortunately not while driving. Your episode involving the Wired article on rabies, however, fell well under my head as I had actually and accidentally read it the week before. I really wish that you had read the article more carefully because you stumbled over a number of important points. There may have been some antivirals involved, but I believe that the article mentioned that ketamine was the primary coma-inducing agent and was also believed to exhibit some antiviral properties. I may have that a little off, but it's close. More importantly, the coma was induced because of the observation that most of the neural damage was caused by overstimulation of the neurons, which in turn causes neural death. I believe it is called excitotoxicity. The drugs used to generate the coma were chosen for their ability to decrease neural activity. The idea was to reduce neural activity long enough to allow the neurons to survive until the immune response kicked in. Most importantly, and related to the second point, the girl was not fine afterwards. She has had to go through months of physiotherapy just to be able to walk and talk again. A year afterwards, she is functioning well, but will probably never be 100%. The coma therapy probably did reduce neural death, but it didn't eliminate it. For this type of therapy to be more effective in the future and thus reduce recovery time, and let's face it, death, better neural receptor agonists will have to be selected, ones that more specifically target the excitotoxicity that rabies induces. Anyway, you probably received a great many emails on the subject, but just in case this one proved helpful in some way, I thought I had better send it. Thanks to all of you for the podcast, and I look forward to the next 100. Well, I did read it, Jeff. We did stumble, but I did read it.
I read it too.
I read it too.
I don't think there was any information about how the girl is doing now. That's why we just.
I've seen a couple of stories about her afterwards, and I did know that there were some subsequent sequelae that she wasn't perfect.
Yeah.
But I think fine is a relative term when you're talking about rabies,
Yeah.
Right.
Versus dead, she's doing great.
The statement that excitotoxicity causes the neural damage, I think, is not correct because I think we don't know.
Right.
So I've looked at a few articles.
That is a leading theory.
It is, but it's also possible that there's some direct damage, right?
Yeah.
So,
Yeah, and I noted this paper that from 1992 where they actually show that ketamine treatment in culture of mouse neurons is able to inhibit rabies virus transcription directly. They don't know the mechanism, but either both ketamine and other drugs, just like ketamine, both show inhibition of transcription. So I think it's a dual hit kind of drug,
Could be.
which is interesting.
but nobody's followed up on that, so we don't know if treating these people had any effect on viral load, right? So
Right.
that would need to be done. So that's a possibility. So anyway, those are good points. So thanks for that. Jeff. Thank you, Jeffrey. Okay. I think that's it, right?
Yep.
All right.
Sure.
So Matt, what's the story with this corona? You want to tell us, want to summarize what went on?
Sure.
I bet you followed it, right?
Yeah, so we have been paying a little bit of attention to it. So the basic story is that, and it actually, the funny thing is it links with Ron Fouché, as you've written about on your blog, who does have his mitts and everything.
Wow, Mitt Romney?
Yeah, exactly. So it basically is that there was, so far it's been 2 patients, but the first one that was identified was from Saudi Arabia, a Saudi Arabian man who developed respiratory illness, and then they resolved when he went back to Qatar. And then at the beginning of, this is in October, and then September, he developed another illness that worsened, and then he was sent to London, where he basically got worse, and they did all the normal respiratory virus screens, and there was nothing identified. And at the time, several samples were sent around, or samples were sent to Ron Fouché somehow in Rotterdam at Erasmus. And along the way, he had hinted that maybe it was a coronavirus. So they did, according to Ron, they did a quick pan-coronavirus PCR and found that it really was a coronavirus, and it was novel. It wasn't, when they sequenced that fragment, it didn't look like anything else that they had seen. And then subsequently, there's been another patient who was earlier in the year, in April, where he had, again, severe respiratory symptoms and renal failure. And that patient actually died, whereas the one in September is still alive, or at least in the ICU, as far as I know. And again, with the similar respiratory symptoms, and they went back and identified that coronavirus and have subsequently sequenced it, and they both look very similar according to what's been published. And just this week, there's a PNAS, or a New England Journal of Medicine report about one of the patients really showing basically everything they know so far, supposedly.
Right. And this is the patient who died.
This is the patient who died, yeah. Exactly.
So that's the Saudi patient who went to London, right?
Right. And so, I think it's the whole thing. It's interesting for me on multiple levels as a virologist, but we work on SARS in the lab. We've worked on bat coronaviruses with Eric Donaldson and Ralph Barrick. And everyone worried at the time that SARS, or from at least the funding standpoint and interest standpoint, that SARS was a one-hit wonder, and it was coronaviruses wouldn't really jump into people and cause any more severe disease. And so on that level, it's really interesting that another coronavirus has jumped from animals into people and been able to cause disease.
So these are both very similar. So they're probably the same virus. Is that the idea?
That's the idea. So far as there's been the complete genome of the first patient who's in the New England Journal paper, that genome has been put on the web and has been called HCOV for human coronavirus -EMC for Erasmus Medical College. So that full genome has been published.
Oh, it wasn't isolated there, though.
I know. I don't know how they worked that out, but you'd think that the patient's initials or something would be in there, but I guess.
Well, EMC is encephalomyocarditis, Talk about corona.
Yeah, well, the HCOV would be the corona part, I guess. But the second patient, the only thing that's been published is 200 base pairs to align it to all the other coronaviruses. And they look very similar. So there's one base pair change out of the 200 base pairs. So 99.5% similarity. And when you actually do the alignments and are shown in these papers, it looks just like two other bat coronaviruses called HKU4 and HKU5 that have been only found in bats and only by sequence. So it's just I think it's amazing that that another coronavirus has jumped from bats to people, or at least presumably bats from people, since the virus that's been isolated from the patients looks very bat-like or bat coronavirus-like. And two, that these two people who at least by epidemiologically kind of investigation, they have not have any contact with each other or even kind of been in the same general area other than, Middle East. And they both seem to have a very similar virus. So
do you think they're both independent infections from a zoonotic reservoir?
I mean, I would say that that's the guess at the moment. There's no way to know. There's no even geographic, kind of close geographic link between the two. So no one really knows where they came from. I know there's several groups out in the Middle East now looking at animals and bats and goats and camels and everything they can find out there that could possibly be reservoirs to try to find where it came from.
This Qatari adult, they think he actually contracted it in Qatar, and he spent some time on a farm with sheep and camels, so they're speculating that maybe that's it.
Right, and when you do these kind of studies, I mean, they've actually done all the epi studies where they've looked at all the close contacts between these two patients, and I think they looked at 62 or 82 different contacts, and none of them are PCR positive or seroconverted at all for this virus. So the CDC and the WHO have not put out any quarantine issues or travel restrictions to the Middle East because it looks like the transmissibility is quite low. One of the other things I should point out is that in Saudi Arabia, everyone was worried about the Hajj, which starts soon, I believe. So you have several million people going to Mecca for religious ceremony. So that will bring in many more people from around the world in contact, and then once they're done the ceremony, then they go back to their original countries. So the fear is that there is a potential spread around the world if this infection really is taking hold. So at the moment, we don't know. But I would say, and what I've talked about my colleagues with, is that I would find it very surprising if the only two people in the world that are infected with this virus both ended up in hospitals where their virus was identified as this coronavirus, and Ron Fouché now has their genomes. So
right.
At that level, I think reasonably it's not possible they're the only two people, but we have yet to find out if anyone else is known to be infected.
Well, I did really appreciate hearing Ron Fouché telling people not to overinterpret the data and panic.
He's learned something.
That was poignant. But it struck me too. This is a lot. In some ways, this is a lot like the case that we talked about. The two cases we talked about a few weeks ago with the tick-borne,
the Heartland virus.
The Heartland virus, where you've got a couple of individuals who, again, in that case, they didn't have any real contact with each other, and they just turned up, and they happened to have apparently the same virus causing apparently the same disease. And I'm wondering if in cases like this, maybe what you're seeing is something that just happens seasonally, some zoonotic infection that there's always been some sort of maybe seasonal risk increase, and we just now have the tools to start picking up these cases. No,
I was going to point that out, is that I think that these things probably have occurred and just been called respiratory viruses or, disease of unknown origin, and just kind of they're treated and someone died and then nothing was identified, right? So with the new sequencing technologies and, more communication of scientists around the world, and more virus hunters, essentially, more scientists interested in this kind of thing, that you're going to see these little, chattering events, as Nathan Wolfe calls them, virus chatter, where you have these little jumps from animal to human, back and forth. And
is that what he calls them? That's great. Virus chatter.
I like that.
Virus chatter. Yeah, it's good. That's what he calls this kind of sampling. So the idea is analogous to SARS, where the idea is that you had, even before the outbreak was noticed, for several years before, there are seropositive individuals in the animal markets in China, in Guangdong, where they looked at blood samples, and there's a percentage of the people in Guangdong province that are positive for SARS, even though it was not identified as a virus at the time. So the idea is that you have these mutational events that allow for jumps to people, but not person-to-person spread, so it doesn't take hold. And not until you get the right mutational set in a virus and the right person that potentially has some other condition or the right connection with the neighboring people, that allows it to jump from person to person. So we might have caught it in the middle of this, or the beginning of this thing, of this jump, or mutational spread.
Or not. Maybe it's just not ready. As you said, he hasn't got
Sure.
the right combination, and maybe this happens all the time.
Yeah.
I mean, I would almost bet it happens all the time. And this is just one of
Yeah.
the times we're going to catch it.
And in the case of SARS, that was just the right mutation, and it took off.
Right. And I mean, if SARS was happening now and no one knew about SARS, and this was our SARS, we essentially caught it at the time before it jumped. And so now that people are looking for it and know that it's there, that something is potentially there, we can look for it and really block or potentially block the transmission events that occur later. Mm-hmm. So, I mean, I think this is just what people in my lab and Ralph's lab and Eric Donaldson and a lot of other, and Eric Delward and even Nathan Wolf have talked about, catching these epidemics early before they happen.
Well, if it's even capable of making that jump, because we don't even know what the correlates of that jump would be.
Sure. Well, that's also one thing we should point out, is that there's no evidence yet, direct evidence, that this virus causes the disease in these two people. Right.
Right.
Yeah.
So
yes.
Mhm.
Both of these people had this disease, and they both had this virus, apparently. And yeah.
Yes. Right. They both had serious respiratory symptoms. They both had renal failure, and they both had this virus. That's all we know.
Right.
So what's interesting is now we have this virus, and if it should ever transmit among people, now we have an early isolate to compare that putative virus with, right?
Exactly right.
Do we have that for SARS?
Well, we kind of do. So what we have for SARS, which is an incredible resource, is there is sequence of coronaviruses or SARS-like coronaviruses from animals all the way through to the human epidemic strain. And so you can actually follow mutation of genes or on the gene scale or genome scale throughout, from animals through the early phase, jumping into people, through the middle phase as it starts to spread, and then the late phase of SARS where it spread around the world. And you can follow these viruses as they evolve and what mutational sets are necessary going from animals to humans or animal to animal, and really start to ask the questions about what mutations are important for spread or for replication in bats versus civet cats versus humans. And you can start to play with these things in the lab to really investigate what the viral correlates are of disease and what the things are important for those kind of things.
So what is needed to... Do we know what's needed to transmit in humans in the case of SARS?
Not necessarily. Not directly. We've done a lot of mutagenesis on the virus. And this is work done by a lot of people in the SARS field, including Ralph Barrick, Constance Sabaro, Stan Perlman. We've helped out with a lot when I was in Ralph's lab and continued in my lab to look at using mouse-adapted SARS virus, which is able to cause severe disease in mice, and then to look at the mutations that are changed in the mouse-adapted virus versus the human virus. Because the human isolate of SARS does not kill mice. It replicates, but they don't die. And what Constance Sabaro did was passage the virus over and over again in mice until you got severe disease. It may sound very familiar to what Ron Fouché did for flu. Now I think the moratorium's over, I can actually talk about that, right?
Well, he didn't even get severe disease.
No. So with
Right.
SARS, with the strain of mice that they used, the human isolate of SARS called Urbani, that will, when you passage it over and over again, you can cause death in biopsy mice, which are little white mice. And then we sequenced that virus. There's 6 amino acid changes in that virus. It took 15 passages in mice to occur in lungs. And if you put those 6 amino acid changes back into mice, or into the recombinant infectious clone of the virus, which is what they did in Ralph Barrick's lab, you can cause the same disease. And now you can start taking those mutations one by one and figuring out which ones are important for lethal disease or certain types of inflammation or certain signaling events in the mouse.
Are they in 6 different proteins?
They are the 2 amino acid changes are in one of the proteins, and the other 4 are in 4 different proteins. And just the 2-cent spin on what the one we showed and what we published was that the spike mutation, so the spike protein of SARS is what binds to a surface protein on cells in the lung called angiotensin converting enzyme 2, or ACE2. And that spike ACE2 interaction is necessary for infection. Mm-hmm. The human virus actually uses ACE2 and grows fine in mice. But one amino acid change in the spike protein of SARS will allow for presumably better binding to the mouse receptor, the mouse ACE2 protein. And if you have the other 5 amino acid changes, but you don't have that one spike mutation, then you don't get lethal disease in mice. But if you only have that one spike mutation, that's not sufficient for disease.
So you need this whole set.
You need the whole set. That's the idea.
Well, clearly, Matt, you should take this new isolate and introduce these changes and see if they do.
Well, so the new coronavirus is very different genetically than SARS. It's in the same group, but it's genetically different. So we can't do the same. We can't do it completely analogously, but there are labs underway where they're going to be doing these things. They're going to do mouse work. They're going to do ferret work. They're going to look at immune response, look at disease, to figure out what the proteins do.
Yeah, and you could certainly passage this new virus in mice or ferrets and see if you can adapt it.
Yes, someone could. Yes.
So you have no plans to work on this new isolate, EMC?
No, we do have very rapid plans to work on that. we are. SARS was Ralph's SARS. This is my SARS, essentially. No, but I think that the SARS researchers and coronaviruses and coronavirologists in general are interested in it both at an NIH interest level, but also every coronavirus that comes out is interesting just to compare it to the other coronaviruses.
Right.
Yeah.
So
didn't you say that you had to do some paperwork to be able to get the virus in the first place, and you were all queuing up with that paperwork? And I don't know if you've gotten it yet, or.
So the virus currently is in Bart Hageman's lab in Erasmus in Rotterdam, and so to get it imported into the U.S., we have to get CDC approval. CDC said we got CDC approval already. CDC said we had to get USDA approval, so we're waiting for USDA approval to come through. I had to get our institutional biosafety committee at the University of Maryland to allow us to do the work and to make sure it's safe. So we're in the midst of getting that. That should be almost done. If we want to do mouse work, which we want to do, we have to get IACUC approval to allow us to do the right mouse experiments and make sure we're doing it okay. So all these things are in the works, and my lab and several other labs are clearly going to be working on this. Hopefully not overlapping in our work, so we can all kind of spend the money wisely. But I think it's going to be an interesting virus to piece apart and to compare it to SARS.
This isolate from the Saudi patient has actually been grown in cell cultures. Exactly. That it grows in vero cells, which is the normal cell line that we grow SARS in. Hopefully there isn't too much adaptation to viros, that it's actually a fairly legitimate sequence that we have that looks like the original isolate. I know a lab is going to make an infectious clone, so we can start doing the mutation analysis that would be interesting to do. Yeah. So, all these things that virologists do to work up the virus, I think are in the works for this new one. So it's going to be interesting. I don't know. Stay tuned, I guess, to see what it looks like and what we can find. Cool. So in the meantime, there was a change in the select agent status of SARS, right?
Yes. This has been my life for the past couple of weeks.
Why is that?
Currently SARS is a BSL-3 pathogen. So we work on it in a special virus containment room, which is the air only goes into the room and out the ceiling, and we have to wear suits with respirators and HEPA filtered air is hoods with only HEPA-filtered air coming into it. And that's been well and good, and we've been working on that since SARS emerged and everyone started working on SARS. As a select agent, there's a whole nother level of paperwork and safety issues that are brought into being by the CDC.
So now it's been added to the select agent list, which means what?
Well, so it means that we have. So the select agent list is basically, I don't know if you want to go over that, but it's a list of viruses, bacteria, and toxins which the CDC and Homeland Security and USDA has some deem, I guess, scary enough in some sense, or at least bioweaponable in another sense, that there should be extra scrutiny on the researchers that use them, and the containment should be at high enough level where all of those individuals, the regulatory bodies, feel secure that you're using it and it's safely and correctly. Mm-hmm. And so what that means is that for any select agent lab, every tube of pathogen in that lab has to be logged and identified in a logbook. The amount that's in the tube, the titer of the virus, the volume has to be logged. Every individual that works in the lab has to go through FBI clearance. So you have to get FBI clearance from everyone that works in that lab. There's extra regulations on shipping select agents from lab to lab. So there's more paperwork and security on that level. So it adds a whole nother level of security and, in my aspect, paperwork that needs to be done.
And then you also have to deal with the dual use research of concern issues, right?
Exactly right. So most of the pathogens on the select agent list are also, the worry is that they can be used for, essentially bioweapon issues. So any of the things that we find for SARS, is there something in the virus that we can find that someone else could use to make the SARS worse, or as more pathogenic? So all those questions come into being.
But you don't have to go to BSL-4 for this, right?
We do not. No, we can stay in BSL-3, which is good. The only big wrinkle which we've been working on lately is that SARS is a positive sense RNA virus, which means that the viral RNA that's produced during infection is itself infectious, theoretically. Mm-hmm. So you should be able, theoretically, can take viral RNA and transfect or electroporate that into a cell and get virus out. It doesn't actually work very well for SARS, but biologically it is possible. So that means that the RNA is now a select agent. Mm-hmm. So we're working on now protocols to be able to allow to take RNA out of the select agent BSL-3 and put it into our BL2 lab to be able to do any transcription, real-time PCR analysis on the RNA of both the cells, both mouse, lung, any tissue, or looking at viral RNA directly. So we're working on all these things now. So it's only been a couple weeks since this has been put on the select agent list.
Yeah, October 5th. So this is published in the Federal Register. We'll put a link to that, and you can see the discussion. They ask for public comment. They make a proposal. So this was done in July 29, 2009.
Right.
And then they solicit comments. And they say, we got 10 comments, all arguing that it should not be added to the select agent list. One of them was from Matt Frieman. No, I'm just kidding.
Well, there was a letter sent around by virologists or coronavirologists that everyone signed. So one of those letters is actually signed by many people.
Yeah. And then they got 3 comments from people who thought it should be added to the list. So 10-3, so they decided to add it to the list.
Well, there's a little more than just a simple vote for the comments.
I know. But they clearly wanted to do this.
Yeah.
And they didn't
So they came up with a rationalization. A rationale.
On one side of it, it makes it harder for us to do work, right? Just in the lab. On the other side, I understand on the principle of why it can be a select agent. So I can't argue that. I can't rationally argue why that there's definitely no reason why it should be a select agent. I think that the main reason why it shouldn't, though, is, or one of the reasons why it shouldn't, is because it's really going to make it harder for labs to work on this virus. And
I was going to my question, do you know if there are people now working on SARS who are just going to say, this is just one more thing, I'm not going to work on this anymore?
Well, I would say for better or worse, there aren't that many SARS, labs that work on live virus around, which I also find it funny that they're making a select agent and there really aren't that many people that have it. I would say that there's there probably is a lot of labs and more companies that actually have SARS samples in their freezer that were working on vaccines or recombinant proteins that they use it for making expression libraries or something that are probably going to get rid of them very quickly. So they won't have to be called a select agent lab. So
or even their RNA preps.
Yes, or RNA, right. I mean, no one really knows about the RNA yet. We're working on that angle. But yeah, I think that there are definitely labs who probably have vials around that are going to hopefully just autoclave them and report to the CDC that they don't have any more in their labs.
Now, they also added 2 other viruses to the select agent list, Chapare and Luho. Is anybody working on those?
I'm sure there are.
Okay.
But I don't know.
I don't know those at all, so I just...
Yeah, Luho, we actually did. It was discovered by the Lipkin lab.
Right.
Hemorrhagic fever.
Oh, that's right. Yeah, it's right. It's hemorrhagic fever one.
Yeah, related to Lassa. I don't know who's working on it. And they took some off. They took herpes B virus off, interestingly. Which is incredibly lethal, right? Not easily transmitted to humans. Person-to-person transmission risk is small. I notice on page 61,087 they misspelled coronaviridae.
Oh, I did not catch that.
Coronarviridae, they spell it.
Well, when you're publishing something in the volume at which the Federal Register comes out, I think a few spelling errors are going to creep in.
A spell checker would pick that up.
Yeah, spell checker should have picked that one up.
It should have.
Well, we wish you luck, Matt.
Thanks.
Well, I like the part of this Federal Register bit where they go into the costs and number of hours and paperwork that's going to be required and the hundreds of hours of paperwork they're adding to your burden. So they have at least thought of that.
I'm glad they're thinking. I mean, we're thinking of it too. Trust me.
Oh, I bet you are.
I mean, the good thing about where we actually at University of Maryland is that there are several other select agent labs here who already work with select agents. So our environmental health and safety officers are used to working with select agents. So it's not as much of a shock to their system as it would be other places, I think. So hopefully it will be a smooth transition, and it really hopefully won't cause that much problems other than a little bit extra paperwork in the lab. But we'll see. We have to get inspected by CDC and do all these things. So, but we actually are in good shape. I can't complain too much. We're in good shape in our facility to be able to jump right through the hoops that CDC gives us. So, hopefully it'll be okay.
If there were vaccines and antivirals, would it not have been a select agent?
I think that's a very good question. One of the things they cite in here is there are not vaccines or antivirals for SARS. There are experimental vaccines and antivirals, but there are none that are FDA approved.
The only agent I see on the list for which there is a vaccine and an antiviral is smallpox. Right. But it's still on the list, but that's fine. But none of the other ones have. I ask because someday you could imagine poliovirus could be a select agent, but there are vaccines and antivirals, so I'm not sure that would make much sense.
Watch out.
Yep, they're coming for you.
It's going to be at least 5 years.
Yeah.
Okay.
Yeah.
Anything
the other ones they removed there's a Rickettsia strains that are removed as well. So I don't know. They are taking some off, which is nice, but I don't. I guess it's good to be in the company of all the other nice pathogens on the list, but we'll see.
Okay, Kathy, did you want to mention something?
Oh, just that my screen went blank, but that they cited one fact in that most of the recent cases have been in laboratorians, and I didn't know that laboratorians was a word. It wasn't in my dictionary, but Alan found it in the Merriam-Webster dictionary.
Yes, it's a rather obscure and seldom used word outside of government documents. I think I've seen it twice in stuff from the CDC, and
that's pretty good.
We are laboratorians.
Yeah,
apparently.
I like it. We need a T-shirt made.
Yeah. Very good.
So,
all right.
If anyone from the CDC is listening, we are very happy with where we are. So I welcome the inspections, and we will be in good shape when you get here.
I think people from CDC do listen, but I don't know.
I'm sure there are at least some people from CDC listening.
Mm-hmm. Matt's a good guy. He plays by the rules. I can tell you that. Yeah. All right.
All right. I need a letter from you then of support.
Sure, no problem. I don't know if it'd do you any good, but I'm happy to write it. All right, that's great. Thank you, Matt. If things develop, we will have you back.
Sure. Absolutely.
I have another paper here that I discovered this week, which I thought was really interesting, and so I wanted to discuss that as well. It's a Cell paper. It's called Pathogenic Simian Immunodeficiency Virus Infection is Associated with Expansion of the Enteric Virome. Huge cast of authors. Well, actually not huge, but a lot. First author is Scott Handley, and some of the labs here that I recognize are David Wang, Dan Baruch, and Herb Virgin, Skip Virgin, Washington University, MIT, Tulane.
Bunch of different institutions.
Bunch of different places, Boston. And this is very interesting. So in AIDS, one of the hallmarks of AIDS is this thing called immune activation. And this is a combination of the virus effects of infection destroying the immune system and also activating it in ways that such that it's out of control. And it's really interesting. It turns out that even people who have been treated with triple therapy, who have AIDS, they have triple antiretroviral therapy, they have a higher death rate than uninfected population, and it's partly believed due to immune activation. Their immune systems are not behaving properly. They have not just destruction of immune systems at every level, but also overactivation. And so people are trying to figure out why that is. So they make a lot of cytokines, in particular TNF-alpha, which can be bad in unregulated quantities, and many other things as well. So this is a relatively recent addition to our understanding of AIDS, this immune activation. So one of the theories is that, so one idea is that when your immune defenses are lowered, a lot of latent infections you have, herpes virus infections, get reactivated. They stimulate the immune system, so that contributes to immune activation. And also there's this notion, and it's supported by a lot of data, that your gut becomes more permeable and bacteria or some kind of product comes into your circulation and contributes. And one of the things that's been shown to come from the gut is lipopolysaccharide, this outer membrane molecule that is recognized by the innate system. So there's a lot of work going towards understanding this. So what this paper does is to use a non-human primate model of AIDS. So they use simian immunodeficiency virus. And it's been shown that in these animals, in the right monkey, so depending on what kind of monkey you infect, you can or cannot get AIDS. So in monkeys that develop AIDS, they get immune activation as well. So they basically took a whole bunch of macaques infected with this IV, and then they had uninfected controls.
Right, so the rhesus macaques get AIDS, the green monkeys don't.
Green monkeys don't, right. For reasons we don't understand, but could be partially related to not getting immune activation in the green monkeys. You know, it's really interesting. The immune activation is the single property that correlates with a bad outcome in AIDS. You know, It's not CD4 counts or viral loads. It's immune activation. It's really interesting. So they basically sequenced the gut microbiome of these monkeys, infected or uninfected, rhesus macaques. So that includes,
and specifically the gut virome.
So they did both, right? They did the virome and the microbiome.
And the microbiome, yeah.
And the virome they define as just non-bacteriophages, right?
Right.
Right.
And they do this by starting with RNA, which is an important feature because a lot of the bacterial microbiome sequence analysis that's done is just done on DNA sequences. Right. But they start here with RNA and convert that to DNA so that then they can do the sequence analysis.
Shouldn't it be bacteriome? Doesn't microbiome mean viruses?
Yes,
also?
It should be the bacteriome and the virome.
That is actually a very interesting point of contention. Okay? So many people are kind of upset that when most microbiologists talk about the microbiome, they're talking about bacteria, fungi, archaea, whatever else is in there. They ignore viruses unless they're bacteriophages. And Eric Delwart in particular, he says that the NIH has this microbiome project, but they're ignoring eukaryotic viruses. And
but it goes even deeper than that, that a lot of times they don't even think of virologists as being microbiologists.
Right. I was going to say, most of the time if you say if you're a microbiologist, virology isn't even included in that. Absolutely.
Yeah, that's ridiculous. We're all in microbiology departments, right?
Yes. Right.
I mean, where else would we get a job? No. So, yeah, that's an interesting point. But microbiome should be all microbes in your gut, right, including viruses.
Right.
Anyway, so in this paper, they looked at, RNA and DNA, I think. They looked at both, right, so they could pick up DNA viruses.
Well, by sequencing the DNA, by sequencing the RNA, but you're right, they did start with DNA as well.
Yeah, they did both. And so they have the bacteria, the microbiome, including... And they don't pay attention to phages here, I think. They've defined the virome as a subset of the metagenome that includes viruses that infect eukaryotic cells.
Right.
And they're not looking at phages. And so they have really, really cool results. They basically discover like 32 brand new, previously undescribed viruses in these... SIV-infected monkeys who had immune activation. And interestingly, very few in the uninfected monkeys. So this is the gut virome. Right. They take feces, right? I think that's what they took out of these animals. Mm-hmm. Which probably isn't the whole story. There's probably stuff bound to the wall that doesn't come out in the feces, but it gives you an idea. I found it really interesting that there were so few viruses in the uninfected monkeys. Previously undescribed viruses, 5 adenoviruses, 3 callices, 1 papilloma, 8 parvoviruses, 7 picobirna-viruses, 7 picornaviruses. Maybe I can get something new to work on, right?
Yeah. Mm-hmm.
And a polyomavirus. Both cohorts of African monkeys. So they did also African green monkeys. Whether or not they had SIV... Remember, in those monkeys, it's not pathogenic. They were free of virus infection.
Just a couple of viruses here and there.
I'm amazed. I would think they would have lots of gut viruses, wouldn't you?
You would think, but
I guess. But these are monkeys that are raised in a controlled habitat, and I don't know. Maybe they're
suppose
they did say they kicked out all the insect and plant viruses, which they thought was from food.
Right,
So,
yes.
I'll bet that those... Yeah, they say they did not differ between SIV-infected and control animals. Right. So those guys do have all those viruses.
Right, so they're eliminating phage, they're eliminating insect and plant viruses that are probably from food, so you should only be left with viruses that would really be infecting these monkeys.
Right. But what's cool is that these are like brand new. They're different from anything else seen. Yeah. It's just in some monkeys in a colony somewhere. Can you imagine if you looked everywhere?
Yes, in the SIV positive rhesus macaques, the ones who do get sick, they're just loaded with all these new viruses.
Right.
All of a sudden.
And Kathy, you probably were happy to see that they even cultured the 5 adenoviruses from these animals.
Yeah, this is amazing.
They're able to grow them. I don't know if they couldn't get anything else to grow, but are ads generally easy to grow out, Kathy?
I guess from this experiment, you have to say yes. Yeah, they're pretty hardy viruses, so it's reasonable to suspect that, they could get them, I suppose.
Anything in the gut has got to be hardy to a certain extent, right?
Yeah.
They also found intestinal pathology, which they say is, characteristic of adenoenteritis.
Mm-hmm.
Suggests that these viruses are doing bad things there. And I think they had some viruses in the serum of some of the animals as well, which is the idea being that these are part of the gut contents that are getting into the blood because the gut barrier is being compromised by infection.
Right.
Right, exactly. I mean, there isn't really immunology in this paper. It's more the virus finding, but I'm sure that's coming next.
Yeah.
And it comes to, we've talked about this before, I think, well, Vince and I have, but does finding the sequence of a virus mean you found a virus?
Yeah.
So they clearly cultured out the adenoviruses, but can they say they found 32 novel viruses, or did they really find 5 and they have sequence of the other 27?
Yeah, I think
Right.
technically, yeah, they don't. You got the
Well,
sequence. It doesn't mean the virus is there, right?
They claim they set their parameters fairly conservatively for recovered viruses, so they found more that I guess they excluded.
Right, or that were just fragments, not enough
Fragments
to
of-
put
yeah.
together.
Right,
into
Right. They needed a
concrete.
certain amount of sequence to say, yeah, we're going to count this as one of our viruses, and then that just doing that totaled up to 32, but there could be more.
Right. No, I mean, I guess I'm being it's semantics. They clearly have 70 to 80% of almost all of the viruses they say they have, or at least genome-wise. But I find it interesting that depends on who you talk to whether you really need to find a culpable virus, or if getting the sequence is good enough.
I agree that you have to have the old standard of, if you want to say they're infected by 32 new viruses, I think you have to show that they're replicating there. Otherwise, you can imagine a number of scenarios where the nucleic acid would just be present, right?
Yeah.
I mean,
Sure.
yesterday on TWiV, we were talking about a study where they looked at the microbiome, the bacteriome of saliva, right? And Elio Schechter said, how do we know these are really bacteria? It could be just DNA or dead bacteria. And that's, so
Sure.
on that side, there's that issue as well. So.
Yeah, I wouldn't go so far as to say you have to culture them, because a lot of these are just not going to be culturable. But I think
you never liked working in the lab, Alan.
Well, no, I'm gonna next say something that would be harder than culturing a lot of these, which is you've got to have some kind of additional evidence that there's an actual infection going on.
Sure.
And
Yeah.
either are these monkeys seroconverting for these things? Can you isolate some proteins? Can you do some pathology and get
Histopathology, staining,
histopathology?
yeah,
Yeah, get virus in cells and. Yeah, I think you're right. You do need a little bit more than just the sequence to really say that they're infecting.
Well, one of the interesting things in the paper, which I've actually talked to Eric Donaldson a lot about when we were doing all of our bat virus work, is how many reads or sequences do you need to know that that's a real virus there? So in their adenovirus culture study, one of the monkeys in animal 40, they have only 60 adenovirus sequences. In 44, they have 138 reads. And in sample 30, they only have 2 adenovirus reads. And they're able to culture virus out of those, even, presumably from the one with only 2 actually adenovirus reads. So the depth is actually not as high as you would imagine for deep sequencing, and you can still find real virus there that grows out. So, a couple reads of a virus may actually be enough to prove that it's really there. And if that's true, I think Eric Donaldson's probably found several hundred viruses from the bats that we didn't report. So,
yeah, because you had too few reads.
yeah, so you have to make a cutoff at what's real and what isn't.
Why would you get a read that I don't understand what it would be if it's not actually a viral sequence, though. If you had 2 reads.
Well, the question is it just homologous to to a phage or to a piece of the human genome? It depends how long the
I
reads
see.
are.
Okay.
So their reads are several hundred base pairs long. So I'm sure in the supplemental data somewhere you can actually find those 2 reads from that one animal that has the adenovirus grow out of it. But so say you have at best case 600 base pairs of adenovirus in that one monkey.
Yeah.
Sure, you would expect it there, but again, could it be, of something they ate? Could it be something else? Monkeys tend to throw poo at each other, so when they're in cages, so there is a little spreading around. There's that. We always had that question about how many reads do you need for it to call it a real sample.
Yeah.
Yeah, and in this case, that's an important point for moving forward with work like this. But what they're doing here, of course, is you're looking at the difference between SIV is there, SIV is not there, and that
Sure.
part of the difference is dramatic.
Yeah.
No, absolutely.
Yeah.
I think it's really amazing. I think it's I've never seen a study where they did this kind of thing before, especially with SIV. So I think it's a great model. It's going to be expensive to keep working on, I think, with all the monkey work. But
Yes.
I'd like to see this done in humans. You could easily take stool, obviously, from people with and without AIDS and see if you get the same sort of pattern, right?
Yeah.
I would imagine that Skip and
They're doing it,
Dave
yeah.
Wong and Dan Baruch have their grant submitted once they got their cell paper.
So
If not, Skip, call me and we'll write it together.
One thing that came up when I was describing this to someone at lunch is, well, were those viruses all there and just showed up?
Yeah, good question.
And I think they answered that to some extent in this section where they talk about doing PCR. So once they have these viruses, they designed PCR assays and went back and said PCR was negative for virus infection in a total of 151 out of 151 cases when the next generation sequencing did not reveal a viral sequence.
Right.
It remains possible that the viruses are present in additional animals but are not detected by PCR or the deep sequencing. So what does that mean? Are those viruses just ordinarily sloughed off and an immune competent animal can take care of them, but in the face of SIV and maybe this immune activation, then these viruses can take hold?
I think that's a good possibility, because I was wondering where they came from, right?
Right.
But if they're there all the time, that could easily explain it,
Yeah, I think that, I would say that the implication is that they're there all the time, just like all the microbiome studies where they've been finding bacteria that no one knew were there until you started looking at 16S profiles. So I would imagine that they're there all the time, and once you have immune suppression from the virus, and then you get a little bit of virus growing out, which causes the gut to be leaky and inflamed, and then you get the immune activation by all the LPS from the other microbiome that's there.
Yeah.
I think it's great.
And in fact, they hypothesize that this could feed this vicious cycle where the immune activation is making the SIV disease worse, and then that in turn is causing more viral infection and so on.
Right.
Yeah.
This could turn out to be really an important thing to target. I think it could be crucial. What I found really interesting is that the bacteria don't change.
Yes.
With AIDS, right?
Right.
I would think it would, right?
Right. Within the limits of everything that they can do. They said that for some of it, they didn't have enough of the bacteriome to actually rule out absolutely no changes in anything. But at the level they could look at it, it didn't look altered.
So does that
with
mean,
all this pathogenesis going on, that does seem kind of weird.
It's weird. Wouldn't you? Does that tell you, if it's true and it holds up? Does that tell you that the community, the bacterial community in your gut, doesn't have to be regulated by immune responses at all? It's fine.
I think other studies have shown the opposite of that.
Yeah.
Right.
Yeah.
That you have a little bit of tweaking the system that keeps the microbiome, the bacterial, I'll call it bacterium from now on, in check, right, in the gut. And there's this play back and forth between what's allowed and what isn't allowed.
Well, certainly the guys who live there, the bacteria who live there, the idea in part is that they prevent pathogenic bacteria from getting hold, right?
Right.
Mm-hmm.
But I just don't understand why these didn't change at all.
Yeah,
This is also fecal material. It's not gut biopsy.
Yeah, it's true.
So that may change too.
Right.
But it may just be that, this would suggest that while the immune system has to be there to keep the gut microbiome kind of from invading the gut, for example, invading the epithelium, the immune system might not really have that much influence on the makeup of the bacterium.
Right. That's what I'm thinking. Yeah.
I think that I don't know, but I think there's a lot of evidence in mice and lab animals that the immune response does modify or can modify that, and the bacteriome and the bacteria, both because due to genetic reasons for the host and also for immune response, they're modified. So I think there's a much more elegant and intimate play between viruses, phage, bacteria that goes on there, and what's evolved with our gut immune response. So I think it's great. It actually, I think you guys have talked to Julie Pfeiffer before.
Mm-hmm.
So she has a polio model, right, where she's looking at polio transversion of gut to CNS and she finds similar things where the immune response and essentially perforation of this epithelial layer changes polio infection rates.
Yeah.
So
actually, we haven't had her on TWiV, but
oh really?
We
you should. She's great.
Yeah.
Yeah.
You discussed her paper, I think, at one point.
That's right. We had that she had a back-to-back paper with a MMTV, mouse memory tumor virus paper. In both cases, you need the gut microbiome for these viruses to replicate.
Right.
And
we should
talked
put
about
her in our TWiV ideas file here.
Yeah.
She's very good. Yeah, it's a great model. And it
We have
uses
a
your
binder
mice,
full of
so come
potential
on.
guests.
At least we have enough to last our lifetimes.
Oh,
Yeah.
yeah.
And she uses your poliovirus receptor mice.
Yep.
Mm-hmm.
I'm glad someone is. So these authors say these data challenge the notion that abnormalities in the intestine in SIV is due to direct effects of the virus. We suggest that immunocompromise during SIV infection is associated with expansion of the virome, and these damage the intestine, as shown for adenoviruses. So they think that the damage can be also contributed by viruses replicating there.
And I mentioned this to MD PhD in our department this morning, and he said, yeah, this enteropathy is a real serious issue in AIDS pathogenesis. So understanding that is also pretty important.
I had not appreciated that at all.
Mm-hmm.
I don't know why. I've seen some lectures by Scott Hammer where he shows these pictures of the gut of an AIDS person, and it looks very clean to me. So I just didn't appreciate that.
Yeah.
At all.
Yeah. To me, one thing is that, I'm not sure what they were looking for when they set out to do this, but it seems like they have just a ton of data here that's not even yet mined. And they do make a point that it will be a major task to select the viral agents to be studied to understand the contribution of the enteric virome to disease pathogenesis.
Well, given the number of different viruses and the diversity of the viruses they find, I mean, this seems like it's just whatever opportunistic infection happens to be around.
Right.
Well, you could start with the ones that grow. The adenoviruses will grow, so you could start with those. Hey, Kathy, there you go.
Yep, that's exactly what the people were telling me this morning.
You and Kathy Collins could collaborate, and
right.
That'd be fun.
I mean, what the microbiome guys would do next would be to treat the monkeys with antibiotics and then see if the virome changes during infection, right? So I wonder how much of the how much LPS you need around to actually cause all of the further immune activation. So I know it'd be interesting to see. I'm sure that's all planned at some level.
Well, but what I was thinking with the corollary would be to be looking at patients on heart versus patients not on heart.
Yes.
At their microbiomes, you mean?
Well, no, they're viromes.
Viromes, yeah.
Right.
Well, I include viruses in that. Because I think you have to look at both, right? Because this is puzzling. There's no change in the bacterial. I'd like to know if that is general and applies to people or what. This is pretty cool. I saw this is a seminal paper. I think it's going to lead to a lot.
Well, and I'm sure there are found experiments, populations of people, and case records that you could look at of AIDS patients on antibiotics. Of course, you could probably do some sort of case control analysis or something there, and see, at least get a first approximation. But this should be a whole new field, and since a lot of it would just be stool samples, it shouldn't be that hard to get to.
Yep.
Right. I wonder what this means for treatment of HIV patients. I was trying to figure out what would be implications for actual treatment at the next step as a therapeutically. And I wasn't really sure, because you don't really want to modulate. You don't want to turn down the immune response because that's essentially helping you at some point, but you don't want to turn it up to block these things because that would hurt you too. So,
I heard a talk this week by a guy, Kevin Tracy, who works on how the innate immune system is controlled by nerves, by the nervous system. And basically TNF can be regulated by the vagus nerve, okay? So in people who make too much TNF, they found that stimulating the vagus drops the level of TNF.
Wow.
I mean, this is all new to me. It turns out that the immune system is totally nervous system regulated. And in fact, lymphocytes are innervated in some parts of you.
Yeah.
Do you know that most of the TNF that you make is made in the spleen? And there's one of the vagal branches that goes right to the spleen and controls it. It's really, really cool. So maybe that's an approach, yeah.
Well, and there's also this huge amount of interaction that goes on in the central nervous system between the immune system and the brain.
Right.
Mm-hmm.
So look for a Nobel Prize in that in the future. I just think that is, and to answer your question, Matt, I think maybe that's an approach to controlling this. Who knows? I mean, they have people with rheumatoid arthritis where they're immune activated, and they put implants in them in their vagus to stimulate the vagus, and this drops down the levels of cytokines, and it reverses their symptoms. Like 6 out of 8 people, their symptoms went away. Yeah. So it's a constant stimulation of the vagus. I don't know if that's good long term, but it got rid of the rheumatoid arthritis.
This sounds. I think I covered a talk by either the same researcher or somebody in the same group a few years ago. This sounds extremely familiar.
Yeah, this guy's Kevin Tracy. He's at a new medical school out on Long Island.
Yeah.
I forgot the name of it.
Yeah, I think he spoke at the New York Academy of Sciences.
Cool stuff. Very, very cool. Yeah. I mean, I had never connected nerves and lymphocytes, I got to tell you. So anyway. All right. Let's do some email. Let's start with Kathy.
Uh-oh. Let me pull that up. Where are we?
Mark?
With Mark? Mark.
Yeah.
Mark writes, I heard a recent TWiV episode this week in which it was mentioned that about 20 cases of hand, foot, and mouth disease are confirmed in Canada per year. Including my 1.5-year-old son's case, I am aware of 5 cases affecting the very young that occurred in Houston, Texas in May 2012. There have likely been many more cases in the area. To reduce the spread of this disease, it is important that parents isolate their children for 2 full weeks after they are back to feeling normal. My son Holden did not seem to be experiencing discomfort while eating, but very cold fluids did help his appetite. By the way, I've been listening to your show for a few weeks now on my Android phone using Double Twist Pro paired to Bluetooth headphones. I listen while playing with my son in the water for about an hour shortly before his bedtime. Both my son and his several-week-old sister were home births facilitated by the well-known nurse midwife. Okay, this is a little bit of a digression. And so then he laments the fact that U.S. insurance companies will not cover the standard method for childbirth in the Nordic countries.
This is real virology, right?
Not so much here. Anyway, so he says the U.S. lags behind many of the most prosperous countries in the world in limiting infant mortality rates, and our medical system that incorporates some of the worst aspects of capitalist and socialist systems is a factor in this issue. Okay, well, and then he says, enough ranting, please keep making science entertaining.
Okay, thank you, Mark.
Thank
I'm
you.
glad you listen. Alan.
Robert writes, does this apply to any situations you've seen? And he sends this quote. This is from Oliver Wendell Holmes Sr., a book called Autocrat of the Breakfast Table, 1858. I shall begin, my friends, with the definition of a pseudoscience. A pseudoscience consists of a nomenclature with a self-adjusting arrangement by which all positive evidence, or such as favors its doctrines, is admitted, and all negative evidence, or such as tells against it, is excluded. It is invariably connected with some lucrative practical application. Its professors and practitioners are usually shrewd people. They are very serious with the public, but wink and laugh a good deal among themselves. The believing multitudes consists of women of both sexes, feeble-minded inquirers, poetical optimists, people who always get cheated in buying horses, philanthropists who insist on hurrying up the millennium, and others of this class, with here and there a clergyman, less frequently a lawyer, very rarely a physician, and almost never a horse jockey or a member of the detective police.
I'm going to say yes.
Yes.
Yes, it does apply to some situations we've seen.
And probably the ones we've talked about on TWiV.
Right.
So thank you.
I'm staying out of it.
Okay. Yeah, you got to keep the CDC happy. Right? Do you have the list of emails in front of you, Matt?
Yes.
Can you take Hannah's?
Sure. Hi, TWiV team. Love your show. Keep up the great work. I was just listening to episode 192 and would like to second the request for a Bioinformatics 101 type episode. You might consider getting someone from the Broad Institute, specifically the Viral Genomics Group, on the show. I'd highly recommend Matt Henn, though he appears to have just left position as Director of Viral Genomics at the Broad, which might rule him out. I think the Institute has a very neat model for scientific collaboration, particularly between more traditional scientists and computer geeks. I'd love to hear a discussion about how the Broad is set up, its advantages and disadvantages, etc., as well as to get insight as the great bioinformatics work they do.
It's a good idea.
Yeah.
That's great.
Yeah. We do need to do the bioinformatics episode.
Yeah.
You have to get someone who can
For our
explain
sake.
explain it, right? Yeah.
Right. Right.
All right, the next one is from Joseph. Hi, guys. My name is Greg. I'm an undergraduate at the University of Manchester. I'm a big fan of your podcasts, and over the last few months, I've set up one of my own. The Life Sciences Podcast. It covers a range of things biological, medical related. As my podcast is in association with my university, they've asked me to get some listener feedback. I was wondering if you've ever carried out a listener survey or have any advice on getting useful feedback from listeners. Well, we did do a listener survey, I don't know, about a year ago with a company, Triple Mojo, who came to us because I knew one of the founders. He had done some advertising on TWiV a while ago. And he had a very long questionnaire, and he just gave us a link for our TWiV listeners, and we got about 1,000 responses. Remember, we pushed everyone to say, we need 1,000, we need 1,000, and for weeks and weeks.
We pestered the heck out of people for weeks, yeah.
And we got 1,000, and that worked out very well, and then he gave us the report. It's now shut down, but I have the report, pie charts, blah, 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 everything is right there. But I'm sure there are similar companies, Greg, so...
well, there's also there's a free site called SurveyMonkey.
That's right. Yeah.
You can set up your own survey, and just put together your questions. Try and phrase them objectively if you can. And you can do multiple choice. You can do rankings, all sorts of different formats they have. Then just the real challenge is getting your listeners to actually fill out the survey.
Yeah, you got to bug them.
Right.
I think for you that would be a good thing to do. You just have to pick your questions so that you get good information back.
Yeah. What do you want to know about your listeners?
I mean, ours was designed for a demographic survey so that a potential advertiser would say, this is who I want to target, right? So It depends what you want to do.
And so, Vincent, was this the survey where you got the information that only about 35% of the TWiV listeners are scientists?
That's correct.
Okay. And so
Which
it wasn't
pretty much
just
tanked our options for getting advertisements from lab equipment suppliers, right?
No, it's not bad. 35% is. So that is I think it's a rough estimate. So it could be higher. I'll tell you why. Let me see if I can find the survey here quickly.
Well, while you're finding it, I would say that it took me personally a while to do the survey because I always listen streaming on my iPhone, and I'm never at the computer. I always listen in the car while I'm driving my kids home from daycare. So
I think I tried doing subliminal suggestions in a few of the episodes.
So the first question was, which most accurately describes your attitude toward purchasing new products? Because they want to know for advertisers, do you buy right away? Do you wait 6 months? Do you wait for second generation? Do you wait several years? So anyway, we had a question, which of the following best describes the work you do? So one was research and development, and that was 37%, and that's where I got my number from. But then the other categories were other, 17%, medical/healthcare, 15%. Then IT information services was 7%, teaching 6%, engineering 4%, technical and support services 2%, and all others 12%. So, in the others, there could be some scientists as well.
And I would add the medical. I would think a lot of people would actually click medical if they're, students or scientists as well.
So it could be as much as half, but still, I think it's amazing that even half of our listeners are not scientists.
That's great.
I think that's what we really, really like, reaching all those people. So anyway, there you go, Greg. Kathy, you're next.
Juliana writes, different animal species have different kinds of placenta and different levels of interaction of fetal maternal tissue and blood. And so then she asks, are trophoblast cells from other species also resistant to viral infection? Can the conditioned medium of human trophoblast cells interfere with animal virus infection of human cells or human viruses infection of animal cells? Thanks. So she's referring to the TWiV that was from ASV this summer, where Carolyn Coyne was one of the guests. And so, Vincent, you wrote to Carolyn, and she wrote back, we have never directly tested non-human placentas for their infectivity or ability to transmit viral resistance to recipient cells via conditioned medium. Presumably, many would have a similar mechanism of resistance to those that we study in the human placenta. The cluster of placental-specific microRNAs that we've been studying and which confer viral resistance are specific to primates, but it is very possible, and perhaps likely, that other animals have specific microRNAs with similar functions. I will note that, in vivo, the mouse placenta seems to be rather impermeable to most viruses. Studies on mouse cytomegalovirus rely on microinjection of the virus directly into the placenta, for example. But there are not many studies that have investigated this question specifically. In terms of animal viruses, we have not specifically tested these. However, given that the mechanism of viral resistance induced in recipient cells seems to be rather nonspecific and far-reaching, parentheses, autophagy primarily, but perhaps others, I would anticipate that many viruses of animal origin would also be susceptible to the antiviral effects of the placental conditioned medium. So, I think that answers the question pretty well. It's nice to have Caroline's expert information on that.
Yeah, I would have, since it's not published, her work, I thought to just send it to her.
Mm-hmm.
Alan, you are next with Clyde.
Clyde writes, "Greetings from North Carolina. I'm a PhD student studying norovirus in the Food Science Department at NC State University. Norovirus, as you probably know, is a difficult virus to research in that there's no culture system available for it. I recently read a paper from a group at Baylor describing a new way to culture enteric viruses. The system works by creating a lab-grown intestinal epithelium from stem cells. The paper shows that the concept works for rotavirus. Perhaps this is a first step toward a functioning norovirus culture system. I thought perhaps you would enjoy the read and maybe entertain the idea of discussing this on a future episode. Thanks for TWiM and TWiV. I'm a big fan of both." And we have a couple of links here.
So
so you got right, Kathy. You asked.
So the primary paper was in MBio, which is an open access journal, and so I don't know if Vincent wants to put a link to that here in the letters or whatever. But it was from Mary Estes' lab, and Stacy Finkbeiner was the first author, and we have one of the two mouse norovirus discoverers in my department. The other one is in Rich Condit's department. In fact, Stephanie Karst was on TWiV, and Christiana Vobis is the one here. And they both were in Skip Virgin's lab, the senior author on the paper we discussed earlier. Anyway, that's connecting everyone up. Christiana had talked to Stacy Finkbeiner, the first author of the paper, because Stacy's now here at the University of Michigan. So in this paper, they used a system where they have organoids. And these are cultures of cells that they used to look at rotaviruses. And like Clyde, they thought maybe this might be a way to grow noroviruses as well. And they tried human noroviruses in parallel with the rotaviruses, evidently, but they didn't have any success. And then Christiana mentioned that at ASV, there was a group that presented in Workshop 31 that the human norovirus gets into these organoids cultures, but to her satisfaction, they didn't demonstrate an increase in viral RNA that would indicate replication. So this might suggest that there's abortive infection. And Christiana is also in the camp that thinks that, as seems to be the case for mouse norovirus, intestinal epithelial cells are not the host of the human norovirus. And since the organoids don't include hematopoietic cells, which might in fact be the host, the organoids might not succeed for noroviruses. And Christiana's final comment was, 40 years of trying to grow the virus in intestinal epithelial cells hasn't worked. So, good idea. They've already tried it and didn't seem to work.
But I just want to point out that these organoids, or a couple of different groups are working on similar systems, are really, really cool. I just recently interviewed Don Ingber up at Harvard, who has a huge grant to develop a series of these. He's already developed a lung on a chip and an intestine on a chip and a couple of other things, and he's going to try and put these together. I think it might be for DARPA, into a system that you could use that would be several organs piped together. He's got a heart on a chip connected to a lung on a chip, and you can study pathogenesis of respiratory viruses in this system, and it's really, really amazing what can be done with these.
Well, that's good. And I wonder looking at the organoids, I heard also from that meeting that the norovirus work was not exact. They couldn't show a huge amount of replication there at all. But if you could start mixing cells in, like if it's known that the hematopoietic cells are important, then if you could start mixing those into the organoid and start making mixed culture systems, and that may be a way to get it to work. I don't know.
Right.
Exactly. Yeah. In fact, one of the things that Ingber has done is he's trying to make a more realistic intestine on a chip, and he did discover that even with just the simpler one, he could co-culture bacteria in it with the cells, which of course putting bacteria into your cell culture on purpose is just really, really scary, but it works. The cells don't die. They keep the bacterial cultures in check, and you get sort of a little miniature intestinal bacteriome developing there.
Yeah,
so you could potentially introduce other cell types and maybe reconstruct the whole organ.
Well, that's amazing.
This would be great for virology.
Yes.
Yeah, because we work in these rather artificial systems, but this could really help us move even more forward. Mm-hmm. Our next email is long, so I think I will stop here and move on to our picks of the week. It's okay with everyone, right?
That's fine.
Sure.
Mm-hmm.
Yes, this is a long email.
All right, Matt, what do you have? Yes, you can have 3 picks, Matt.
Is that okay? So
Sure.
I picked 3. I didn't know when I was going to be on next, so I figure I can throw my picks in now.
Can be on any time you want, man.
Well, so 3 picks. So the first pick is for other podcast listeners. So people who probably listen to TWiV also listen to Radiolab, I would imagine. But on the recent Radiolab, it was called Dark Side of the Earth, and it was about they had a really good story by an astronaut talking about being in space and what it feels like to be in space. And it was really an amazing episode to listen to this guy who did spacewalks basically as I can't remember the guy's name. I can probably pull up his name, but who did spacewalks and is talking about him being in the darkness of dark in space. His name's Dave Wolf, and what it felt like and how it made him feel as a human, and it was just, I don't know, the music that goes along with it is great. It's very good.
And it was scary. I listened to it. There's a part where they pretty much got locked out of the space station.
Right. So he tells this whole story about they try to get back in, him and his Russian comrade on the Mir, and they try to get back in and they can't open the door to the spaceship.
The transcript makes it not sound as dangerous as it was. If you listen to it, anyway, we don't want to give away the whole story. You just have to go listen.
It was just really great. It's an amazing story, and the astronaut Dave Wolf is an amazing storyteller, I think. So it actually translates really well to radio or to podcast. So that was the first pick. The second pick is something I came across that was, I would say, is for grad students, and so it was this a blog called Neurochambers, and it's basically the title of the blog is An Insensitive Guide to Striving in Your PhD, or Thriving in Your PhD, sorry. And it's basically 42 points that this author thinks are important to have a really good and successful PhD. So I would say for anyone out there looking to go to grad school or in the middle of grad school, it's worth a look. And my favorite one is the last one, which as a PI I think is very important. And even when I was in grad school was what I worked or strive for. And it says, above all, remember that you and your supervisor are in this together. Those 3 years or more, depending on where you are, can be an energizing, productive, and career-making partnership, but they can also be a frustrating waste of time and energy. If you want your supervisor to go above and beyond for you, then lead by example and work your butt off. So I think that the whole list bodes well if anyone follows that as their kind of grad school mantra.
Yeah.
And the last one is kind of different than what we usually do on TWiV. I don't know if anyone listened to this, any of you guys listen to it, but there's someone who I've seen online for a long time. His name's Gary Vaynerchuk, and he owns a wine company, and he's a social media kind of guru guy. But he's basically a giant motivator. And if you listen to any of his videos, which he has a lot on the web of him giving keynotes at all different kind of big Fortune 500 and Fortune 100 companies, their kind of big giant meetings, you leave it and you kind of want to start a business right away. And I think it actually translates to working being a PI and being a scientist really well. So this one I think I put up was a longer one. There's a lot of shorter ones. So if you just Google Gary Vaynerchuk or listen to this, the one YouTube video I linked, I think he's pretty amazing. And you leave it and you think that you can, you by hard work and by dedication, and by really finding what you want to do in life. You have to find that that one thing that keeps you going. And if you're a scientist, if it really is science, then it matters to find that one thing. And the reason I put it up here was one of the things that I actually emailed Vince about that Mark Chalberg talked about in last week's TWiV, which was when you asked him what advice do you have for young scientists or people who want to go into science, and he said very blatantly, if you wake up in the morning and think you can do anything else in your life that will make you be happy, then go do it.
Right.
And the point being, it's not an easy gig, and it's very fun if you love doing science, but it's not easy, and you have to really have your heart in the right place and have your intentions directed if you're going to be successful. So altogether, I think it's interesting, and people should look.
Nice. Those are great. Thanks.
Mm-hmm.
Yeah, I particularly like that list of things for thriving in your PhD. That's pretty cool.
Mm-hmm.
Cool.
Alan, what do you have?
I only have one pick.
That's fine. It's usually what we all have.
Yeah, I noticed everybody else has a couple of links, but this is a wind map of. I apologize to our international listeners that this only happens to cover the United States and only the continental U.S. at that. But it's just a really cool graphic that pulls data from the National Digital Forecast Database. So this is weather data from all over the country, and it plots these lines based on the wind speed in different locations, and you can watch the wind blow across the country. I find it mesmerizing.
And it turns out, when I first saw this, I thought, did I forget to pick this? But I did pick it. And what I learned when we were doing TWiV is that when you click on it, you zoom in.
Yes.
Which I didn't know till then. But
when did you pick this?
I think you weren't on that show.
It was just Kathy and me.
I missed that one.
Yeah. But, and then you have to click unzoom to get back out. But and it's always different. It's
very cool.
Yeah, very soothing.
And what this shows is what you said at the beginning. You were claiming that your rain was coming from
Ann Arbor,
Michigan. Yeah, it's really coming from Baltimore if you look.
Yeah.
No bad things come from Baltimore.
Sorry about that.
That's okay. We can do it twice. That's fine. Kathy, what do you have?
So I have this one. How much does a shadow weigh? It's a YouTube video, and it tells you that light actually has weight. So on a sunny day, Chicago weighs 300 pounds more than on a cloudy day. And therefore, if you want to weigh yourself and you want to be less weighty, you should weigh yourself in the dark. And it talks all about the physics of this in a way that I could pretty much follow it. And then toward the end of it, he starts talking about these slow motion slinky drops from Veritasium. And his first link is to one of those slow-mo slinky drop YouTubes. And when you start watching that one, there's links within that YouTube to even slower mo ones, and it's just totally fascinating.
These are
really cool physics.
Really cool.
Yeah. So I just got lost in the physics stuff when I was picking these.
Yeah, your yo-yo pick last time was very popular.
Yeah.
A lot of people commented on that.
Yeah. Well, check out the slow motion slinky stuff. It will blow your mind.
I can't watch it while I'm...
No.
Because they start to play right here. I get that autoplay, but yeah, they're cool. I have two picks. They're both short. One is an article by Ed Yong. It's called The Parasite's Parasite. It's about a new large virus and a virophage recently discovered in France. And I had an interesting correspondence with Ed this week because he had subtitled it "A Virus That Infects a Virus." And I said, no, Ed, it cannot be. And he was good enough to change it. And he agreed that you can't say a virus infects a virus. It's actually a virus infects a virus-infected cell, of course. Anyway, it's a nice summary of that. And we'll probably do it at some point here on TWiV because it's a cool story. The title of the paper is "Provirophages and Transpovirons as the Diverse Mobilome of Giant Viruses." These guys make up all kinds of words.
Mobilome. That's
Mobilome. There has also been a similar article in Ars Technica by John Timmer. Is that his name? Can't remember. Do you know that writer, Alan?
I don't think so.
Let me see.
Not that means much.
John, no, that's not it. Ars Technica. Let me find it. Ars Technica, John. I want to get it right. Yeah, John Timmer. And he also said virus infecting a virus. So I corrected him, and he fixed it too. So I have a very fulfilling week here.
There you go.
And
correcting the internet one at a time.
Yes.
They listened, which is good. They didn't say you're full of it. No, I'm going to leave it. They were very good about it. So thank you. And there's the article. And then I also found the very interesting post this week called "All the Cool Kids Are on the Archive and Haldane Civ and Why You Should Be Too." So have we mentioned Archive before, Alan? I think we have, right?
I think we might have in one of the discussions about open access publishing.
So this is a server that many physicists use to put their papers on, and now biologists are getting into the act. And it is a place you can put your manuscript before it's submitted for publication. It doesn't prevent you from submitting your manuscript normally. And you actually get a date stamp, and you can say, I published it first on Archive and so forth. So there's an interesting post about why it's gaining traction in biology. So I thought it would be of interest because we talk about open access a lot here. I'm just wondering if sending my next manuscript there as well.
How does it not break copyright for a journal that you're paying to publish? They know that you're publishing in.
Alan?
Well, that's a darn good question. So in physics, this has been the standard procedure for years, that it derives from this tradition of mathematicians mailing their results around to each other, and then Archive kind of grew out of that, and actually the World Wide Web kind of grew out of Archive. So this goes way back in physics, and physics journals have already made their peace with republishing stuff that was already on Archive, and I don't know exactly legally how they handle the copyright aspects. One thing about copyright, though, is you can't copyright information. You only copyright a particular presentation of the information.
Right.
So it may simply be that the paper is different enough by the time it's published that it's a separate thing. Of course, open access journals don't have this problem because you retain your own copyright and it stays with the researcher the whole time. How the biology journals and the biology sections of journals like Nature and Science are going to handle this, I don't know.
Well, there is a web page which lists the policies of Nature, Oxford, Elsevier, Springer, Wiley, PLOS, and they all say you can do this.
Really?
Right.
I put the link in the show notes.
Interesting.
So PLOS, for example, Nature says if scientists wish to display drafts of their research papers on an established preprint server before or during submission to Nature or any Nature journal, that's fine by us.
Yeah, and they put that in there for the physicists. I don't think that they anticipated that the biologists would follow.
Yeah, so they may change, huh?
Yeah, because, frankly, the biology is where the big money is.
I see.
That's where there are many, many more journals, and a company like Elsevier makes most of its money off selling overpriced subscriptions. Selling subscriptions to biology journals that are...
so this is going to be a developing story then.
This is definitely going to be a developing story.
That's interesting. I guess who's going to be the first one to have their science paper rejected because they put it on Archive instead of...
It's not going to be me. It won't be me, I can assure you. Because I wouldn't have a science paper. Let's see. Anyone else? No, that's everyone. Okay, we have a listener pick of the week from Niseen, our friend down at Gainesville in Rich's lab. Hi, TWivers. I was changing channels on my TV, and I found this movie on TMC, Jungle Gym 1948, with Johnny Weissmuller, Virginia Gray, and George Reeves. Lady scientist Hilary Parker is searching for a rare drug to help combat polio. Opportunist Bruce Edwards joins the quest, but is actually after gold and buried treasure. The movie was made before most of your listeners were born. It was made before I was born, too. Mm-hmm. And everyone on this episode, for sure. Cool. I didn't know that. A cure for a rare drug to help combat polio. Well, I guess.
And in 1948, that was worth seeking.
That's pretty good.
Yep. Okay, and that will do it for our picks and for TWiV 204, which will be at iTunes, TWiV.tv, and the Zune Marketplace as usual. And if you like TWiV, one of the great things you can do is to go over to iTunes and find it there, which you can do by searching my name or This Week in Virology, and just rate it, and that helps to keep us visible. Do send us your comments and questions to twiv@twiv.tv. Matt Frieman is at the University of Maryland in Baltimore. Thanks for joining us today, Matt.
My pleasure. Thanks for having me.
Always good to hear about coronaviruses. Kathy Spindler is at the University of Michigan, Ann Arbor. Thank you, Kathy.
Thank you. As always, this is a lot of fun.
Great fun. Alan Dove is in Western Massachusetts, and alandove.com. Thank you, Alan.
Always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.